Yossi Paltiel. I am the head of the Institute, the head of the Prof Lab, and most importantly, the head of the Quantum Nanoengineering Laboratory. In our lab, we are trying to take or understand biological meaning or try to see biological processes, see if there are quantum processes in those biological processes, and take them to build devices, quantum and nano devices. So we start with biology, basic question about quantum biology, understand those questions, and then do transport and electrical measurement, seeing why it is important for biology, going to application at the end. So I would like to give one small example of how things going. We started asking why nature is homochiral. Why in nature we have only one chirality in nature, why the symmetry is broken, and we found out that this answer relates to spin. Wherever we pass current through chiral molecules, only one electron spin will go through. So this was starting by a basic question in biology. We found a mechanism in physics, and we can understand why it's important for biology, because we reduce scattering, we preserve angular momentum. So that's why every time charges go through protein, we don't want the protein to be conductive, but we want the protein to be immune against external fluctuation. So this is, we want the biology to be stable, homeostasis, against changing external condition. So let's start with this biological process, why it is important for physics, because there we can use it in a very simple way to do spintronic devices. We can have very high spin polarization by very small coating of chiral molecules. Another thing is, if you have more than one spin going through those kind of layers, like in respiration in biology, or photosynthesis, where we need the ox to have the oxidation, where we need two electrons with the same spin because the oxide ground state is a triplet. Again, we can understand why it is important for biology to be chiral, because we want the surface to preserve spin and to have two entangled electrons going through. Why it is important, again, to energy, because whenever we split water, doing the same as photosynthesis or in biology, wherever we do that, we think we want the process to be very efficient with no side products. So this is why if we align the spin in chiral potential, we see those kind of things. But we can also use it for energy, because wherever we split water, the hydrogen is at singlet state, but the oxygen is in a triplet state, so we have a more efficient energetical process with no over potential. So we started with a very basic questions about quantum biology, ending with application in physics for spintronics, and also having green energy, where we have few startup companies. We are combining together physics, biology, and chemistry, starting from basic scientific question, ending with application, and our group is very diverse. Some of the students are working on superconductors, or the basic questions. So for example, superconductors with chiral molecules. Others working on rather immediate application, like batteries or improving batteries and green energy.
My name is Yael. I'm a PhD student in Yossi Paltiel-The lab in the Applied Physics Institute in the Hebrew University. In my research, I explore the idea of using organic molecules in magnetic memory devices. The organic molecules is an easy and abundant and cheap way to control the magnetism of the magnetic memory device. Using this microscope, I'm able to directly image the magnetic domains of the device, and I'm able to see the effects of the chiral molecules on the domains. The domains are the internal magnetism of the materials.
This is a lab where we do the basic spintronic devices, superconducting spintronic devices. We study the devices with chiral coating, where we add the triplet state to the superconductivity and the spintronics through chiral molecules to standard devices. We measure the devices going from a room temperature down to 300 millikelvin with a gradient magnets up to 9 Tesla.
is Ofek Vardi. I'm a PhD student at the Applied Physics Department, and I'm studying nuclear spin effects in biological systems. We have just recently published a paper in PNAS discovering new nuclear spin effects in biological systems, specifically water entrance into human cells. I'm also studying the interactions between spins inside of proteins to explore how they interact and how they affect the protein folding process and how they help with the protein structural stability. I'm also studying how the spin-spin interactions in proteins affect their folding process and their structural stability. So we were able to observe nuclear spin effects using 3 oxygen isotopes inside of water molecules, and we looked at human cells and how the water molecules enter the cell, and we were able to measure differences in the isotopic ratios indicating nuclear effects in the process of water entering the cells. We were also able to observe very distinctive differences in the water splitting process when we used different isotopes of the water, like we did with the cells. So all in all, both chemical and biological processes were affected by the nuclear spin inside of the process.